Okay, welcome back to the symposium about parallel computing and quantum devices. It is my pleasure to introduce now the next speaker for today's session, Professor Greg Engel from the University of Chicago. Greg will present us a talk about quantum dynamics for real-time processing of excitonic energy, tuning Vibronic coupling to steer energy transfer within a photosynthetic complex. Please, Greg, stage is yours.
Thank you so much. It's a pleasure to be here with all of you today from afar in Chicago. I really appreciate the invitation from Rafi, Yossi, and Elisabetta. So I'm going to speak today about quantum dynamics in photosynthetic systems, and something we are really excited that we recently saw in some of our two-dimensional electronic spectroscopy, that we see a change in redox conditions that changes the energy flow through the system in real time in response to oxygen. And we're excited about this because there have been so many questions about the roles of coherence and vibronic coupling and other elements of energy transfer in these systems. And now for the first time, we have a handle. So in my group, we want to look at how to harvest sunlight. We want to think about how to reach out and grab that photon, use that energy to do something productive. And where we like to look for inspiration, though not the devices themselves necessarily, where we like to look for inspiration is biology. We like to look at photosynthesis. There's been about 2.4 billion years of R&D as evolution has worked to optimize this process, and it is indeed highly optimized. So we want to look at this light harvesting initial event of photosynthesis. So from this middle school textbook, explaining the biochemistry of photosynthesis, the part that I'm going to talk to you about today is the little orange arrow at the bottom, just this little bit right here, of how you move from an antenna, this distributed source of excitation in order to capture the sunlight, and then move that energy to a reaction center where charge separation happens. I would note for the engineering audience, this is a very different strategy than what we typically see in our own devices, where we use the same materials to harvest light as we do to separate the exciton into an electron and a hole. Biology does it very differently. They create this big antenna and they funnel the energy to a single place where that charge separation happens. I find that actually really exciting when one thinks about optics and cameras and pixels as well. So I think there's a lot we can learn from this. But biology also takes a very engineering approach to this problem that I want to highlight early on because it'll be important as we think about the environment. They use bacteria chlorophyll or chlorophyll for almost all roles inside photosynthesis. It does the light harvesting, it enables photoprotection, it does energy transfer, it separates the charges in strongly coupled bacteria chlorophyll molecules, and it does electron transfer as well. It's a really exciting approach to not build a new molecule for every one of these tasks, which is what I as a chemist would have done, but rather to use the same molecule over and over again in different environments in different ways with different patterns of couplings and geometries in order to power photosynthesis. And what that tells you is the environment around the system is absolutely crucial to the dynamics. And when we start thinking about quantum dynamics, this notion of dephasing and the role of the environment becomes absolutely critical. And that's one of the things we like to keep our eye on in my group to try and understand. So the bug that I'm going to talk to you about today is Chlorobacter tepidum. It's a green sulfur bacteria. It's one of these ancient ones from about 2.5 billion years ago. It's still around. It lives in the bottom of microbial mats under an inch of sludge or at the bottom of the Black Sea. It doesn't live in desirable environments. It has a doubling time in some of these light-starved environments of about 22 years, which is roughly our doubling time. It's not the typical life of a bacteria. It's not a good life. But they scrape out an existence, and the way they do it is by being very efficient and never dropping the energy from their photon. They're very, very good at light harvesting, and that's what they've evolved to do, to be robust and to transfer that light with high efficiency so that they can avoid damage and harvest as much light as they possibly can in these very dark, undesirable environments for a photosynthetic organism. And the particular complex I want to talk to you about today is the Fenna-Matthews-Olson complex. It's a little protein that's just kind of a connector. It's a wire. It's, from a computing standpoint, this is your bus wire. This is not where the action is. but it has to work for the whole system to work. It's what holds it together. It connects the antenna to the reaction center. And the inside there are a series of 7 bacterial chlorophylls, and they're arranged, as you can see on the screen, in what looks a little bit like a jumble, but they sit inside this kind of taco shell of protein, and they couple to one another. And so when we look at these, what I see as a spectroscopist are these 7 chlorine rings, each of which have a strong transition dipole moment, and I see 7 two-state systems. So for people who like to think about quantum computing, these are 7 coupled two-state systems, and that's a reasonable description. And because they're coupled, the next thing you do is diagonalize that Hamiltonian. You find out that the excited states are somewhat delocalized. Just by the nature of the Hamiltonian, the lowest energy exciton near the reaction center is quite localized. That's how it transfers out. The higher energy on the other side is where energy comes in from the antenna, and it flows through. And Tobias Brixner and Graham Fleming did a really seminal study of this protein in 2004 with two-dimensional electron spectroscopy, looking at the pathways through which energy flows. What I want to look at here is not so much the how, but the why. What is it that drives the energy in these particular ways? Is there a way to change it? Is there a way to understand what's going on? And can we see the microscopic details of why the energy transfer is so efficient so that we can extract design principles? And that's my real interest in this particular molecule. So there were a number of studies that came out thinking about what was going on inside this complex. So there's an early paper with Graham Fleming where we saw interesting coherences and quantum beats in our signals. There was a follow-up work looking at how the protein protected these. We were able to see that this persists up to physiological temperature with our work in PNAS and Elisabetta Collini's work in Nature. She scooped us by about a month. But the important part, I think, from all of this work, actually, is the ideas that came out of it. So Martin Plenio, Susanna Huelga, Alana Spiru-Guzik came up with ideas looking at environmentally assisted quantum transport and noise-assisted quantum transport and ways to move energy that exploit the dephasing and whether this could actually be functional. And recently in cold ion-trapped systems, I believe in Austria, there was a 10-qubit system that implemented this and shows that this algorithm really does work. And it's a quantum random walk that does get a little bit faster with dephasing. And so there's exciting ideas that come out of this, but there have been a lot of questions. I mean, a lot of people attacked our early papers and said, is it real? Is it right? Are you looking at vibrations? Are you looking at electronic coherences as you claim? Or is it much, much more complex where the two mix and lose their identity and are actually vibronic, where the electronic transitions couple to the vibrational transitions? And this created a lot of interest and a lot of controversy. And ultimately, what we need is data. We need ways to tune this and see what's going on. So, the first papers that started to come out with this were thinking about non-adiabatic effects inside these complexes. And there's a beautiful work by Vivek Tiwari and David Jonas looking at an avoided conical intersection and the particular dynamics for non-adiabatic mixing. There were papers by Susanna Huelga and Martin Plenio, and I want to highlight this idea that there's strong coupling, and the excitons have actually evolved to match their energy gaps to the vibrations, precisely so that they can dump energy into those vibrations. And this is something where Alexandra Alea Castro and others have really thought about the classicality and non-classicality of energy transfer dynamics in these. And she has a whole series of beautiful papers looking at exactly the microscopic elements. But for this talk, I want to highlight this idea that the excitonic system may be tuned to the vibrations, because that was a key observation by Tiwari and Jonas as well. And then there are other papers looking at the characterizing these as electronic or vibrational. There are neat papers looking at the mixing of these and what happens if you go to a Vibronic model where you couple, in this case through simple Coulomb couplings between vibrational electronic states, and you start to see complex mixing. There are lots of these papers, but there was another series of breakthroughs that are equally important for this talk, which is getting genetics in these organisms. So this may sound very basic, that of course molecular biology has genetic capabilities now, has for 100 years, but for photosynthetics, you have to do this in the native organism because of all the chaperones and the attendant biosynthesis. And for green sulfur bacteria that live only on sunlight, they don't take carbon from any other source. if you knock out the photosynthetic apparatus or any piece of it, you kill off the organism. It's a fatal mutation. And so getting genetics in these has been a 30-year project. And just a few years ago, Bob Blankenship and his group were successful in doing that. And they started creating some mutants where they could test ideas. And one of the things they looked at was what happens when you expose these complexes to a little bit of oxygen. And actually, it's just a very little bit of oxygen. Just handling in air is enough. And it turns out that there are a couple of cysteines that oxidize and they become thiol radicals. They can't form a disulfide. They're too far apart. They form these thiol radicals, and this changes the fluorescence lifetime. And this is something that Bob had seen in the early 90s, but didn't understand. And now with his mutants, he can see. And then there are other groups that started looking at various mutants to knock out different chlorophylls and look to see if these vibrations were robust, or these quantum beats were robust. And they really saw that it didn't seem to matter. And then we started looking at those same cysteine mutants, the ones that are actually show a change in function. And we started seeing very different patterns of the energy transfer through the FMO complex. And this was exciting because for years we've tried to engineer different systems that might show similar character or not, but we've never been able to break this model, leading us to, either believe it's quite robust, which evolution does do sometimes, or perhaps that we're looking at the wrong thing, which scientists do sometimes. And so we wanted to try to resolve this with data and see what was going on. And now for the first time, we had an easy, simple handle. You expose it to a little bit of air, you see one thing, you add a little bit of dithionite or ascorbate, you see something else. And this was really exciting. So let me go and show you what this complex looks like again. Here are 8 bacterial chlorophylls in the complex. I know I told you there were 7 before, but it turns out if you handle it really carefully, this one at the top doesn't fall out. And that's something that we learned around 2010. And so our new samples definitely have 8 chromophores. The 2 things shown in red, here and here, are the cysteine residues. They're right near the bottom where that reaction center sits, where the energy would be funneled to the reaction center. That's where the 2 thiol radicals form. And then at the very bottom of your screen, you'll see something in orange, which is a tyrosine tryptophan chain. Harry Gray and others have seen these in many electron transfer enzymes, and they can move a charge out of the active site of an enzyme. And here what we found is after we mutated out the 2 cysteines, which we thought were to be really the only redox active residues, because most of the tryptophans are bound to the magnesium in the bacterial chlorophyll, we found this tyrosine tryptophan chain that still could be oxidized and still affected some of the energy transfer, especially the things down near that bottom bacterial chlorophyll. So this was exciting to us. And we realized in some sense what might be going on, that if Bob has really found a photoprotection mechanism and you have to dump this exciton, either by letting it quench and recombine or by removing a charge from the system, this chain may be functional. And this was something we started thinking a lot about. And I want to show you a chemist's view of the electronic structure inside this context, now that you're oriented with the reaction center on the lower left, this tyrosine tryptophan chain, and the 2 cysteines. So exciton 4 sits kind of in the middle of that taco shell. Hopefully you can see the bright chromophore. It's pretty localized, but you'll see if you look very carefully at your screen, some fainter chromophores down here and over here, and it's a little bit delocalized. Exciton 2 sits down here, and it also localizes on these same sets of chromophores. And then exciton 1 is really quite localized here. And you do see there's overlap. That's how a Redfield mechanism will move energy through these complexes. But you can kind of see what happens if you mutate these 2 cysteines, how the energy transfer is going to change, and which exciton's excited states, that is, it's going to affect. And so we can go through and we can try and understand what's happening. So we do that. First, we look at the linear spectra because it's always a good place to start. And we look at the wild type. We look at what happens if we mutate cysteine 353 to an alanine, or cysteine 49 to an alanine, or if we do the double mutant and mutate both. And what you see here, aside from a little bit of scatter background up here, which actually catches the eye more than I wish it did, the pattern is really quite similar across. There's no large-scale rearrangement of the excitonic structure. Instead, they're very small shifts. You can see that exciton 1 shifts a little bit here. You can see it shifts a little bit more there. Interestingly, in the double exciton, it shifts very, very little, which is unusual. And we're going to see a lot of these non-linear or non-monotonic effects. And that's something I want you to pay attention to throughout the rest of the talk. And let me give you the result first, because I think it'll help you understand some of the spectroscopy. What we see is in the wild type, in a reduced environment, we see the energy basically go from exciton 4 straight to exciton 1. Or excuse me, through exciton 2 to exciton 1. I apologize. And only 13%, I'm sorry, I'm failing with my slides. In the reduced environment, we see half the energy go through exciton 2 and half the energy go through exciton to exciton 1 directly. In the oxidized environment, we see it almost all go through exciton 2. It bypasses that direct channel and goes around near the tyrosine tryptophan chain. Now, for close readers of Martin Plenio and Suzanne Huelga's work, this idea that you would have what they would have called a balanced interferometer is very interesting from an interference standpoint and a coherence standpoint. And then when you break that and you detour all the energy through this exciton near this quenching chain, it shows you how you're going to quench the fluorescence. And so we understand what's happening. But now for the first time, we kind of have a handle on what's going on. And I want to show you why we're confident in this and why we attribute it to a Vibronic model that's going to affect the dynamics inside this complex. So we look at our two-dimensional electronic spectra. And for those who are practiced, you're going to see some very subtle differences. You're going to see by a picosecond how here this looks homogeneously broadened. This one looks tilted. It looks inhomogeneously broadened. You're going to see different intensity patterns and different structures throughout. And importantly, what we have, I think even more important than this system bath element you see in the line shape, is actually the time evolution and the evolution of all of these features and the rate constants of energy transfer throughout and around this two-dimensional spectrum. And my student, Jake Higgins, who was looking at this, he was pulling this apart, and he was very clever. He realized that we have all of the perturbation theory diagrams and the Feynman diagrams, and the signals are always attributable to linear combinations of these diagrams, basically interferences between different perturbation pathways. But the fundamental processes that we're looking at shouldn't be attributed to a given peak, but they end up being attributed to linear combinations of peaks. And in doing this, he started looking at what Ham and Zani call dynamic Feynman pathways, those that change with time. So certain things like a ground state bleach, it just sits there. But then you can create a bleach recovery signal, which will cause your ground state bleach signal to change. But this dynamic Feynman pathway is the one that changes with time. And by looking at this a little differently, it was really foreign to me as a avid reader of Shaul Mukamel's book to think about things in this way. But it turned out to be really constructive in isolating the particular population dynamics that we wanted to between the diagonal and the off diagonal, and then building up a way to look at multiple rate constants at once. And so we did this. We did this on the diagonal features. We did this on the cross peaks. We looked at these different things. And then we found that we could take linear combinations and recover the fundamental dynamics of, say, a relaxation rate between two states, which we can then separate from the ground state bleach recovery, or the difference between losing things through a channel that goes through exciton 2 versus directly to exciton 1. And this was exciting to us. When we did this in the experiment, we found some really unusual results. We found some things that made sense. Like, for example, if I just have you focus on this middle column here in the reduced system, you have a fairly fast 500 femtosecond time constant. If you make a mutation to 353, it gets really slow. You make a mutation to C49, doesn't do anything. And of course, if you make both, you see what you would expect, that it gets slow. One affects it, one doesn't. No problem at all. Then you come down to, say, the oxidized dynamics in the same middle column, and look at this. If you make a mutation to 353, the rate gets faster. Okay. If you make a mutation to C49, the rate gets even faster. And if you make a mutation to both, it gets marginally slower. That's an unusual type of effect. That's what I mean by a non-monotonic effect. This is why we thought there was an opportunity to learn something. That if one mutation goes faster and then a second mutation goes faster, you would expect naively that both mutations would go even faster yet, and it's exactly the opposite. And we see a lot of these types of patterns within the data where we see some unusual dynamics. And we wanted to go in and try to understand it, so we built a model. We built a very simple red field model, and we found, and the model, I should say, was copied from Adam Kell and Rizard Jankowiak in Kansas. And what we found is the only way that our model could match our data is if we took their spectral density and we added an extra vibration. Here it's just a Gaussian, it's an incoherent vibration in the spectral density. And we found some interesting things. First of all, that we found that we could come up with a consistent pattern where if you oxidize one, so you go from a wild type reduced to wild type oxidized, and then as you change the residues, you can kind of chart out how you move. So if you mutate C49, you come down. If you mutate 353, you move to the left, or you can go in the opposite order and get to the double mutant. And we sort of put this on the background of the rate constants we would expect from our model. And we found that we had a lot of these types of nonlinear things. So here you see a red stripe with blue on both sides. And you can kind of see how if you move from wild type to oxidized, which causes a site energy change, and that happens because of the thioradicals. If you remove the thioradicals, you move back down towards where you were. If you remove both, you're similar to where you were before. You're roughly at the same energy level, a little bit below. But interestingly, you've really changed the system-bath interaction because you've given a little more motion to the bath by making these mutations that are a little less bulky. And all of a sudden, you move from a blue stripe through the red stripe and back to a blue stripe, and you see these non-monotonic behaviors. And what this is coming from is the fact that you're shifting the inhomogeneously broadened array of exciton energies away from the resonant vibration. So here the vibration is relatively narrow, the distribution of energies is relatively broad, but there's enough of a shift if you move it about 100 wave numbers that you move it off of that resonance. Only maybe a little bit of the tail is there. And this explains the change in rate constants, it explains the change in branching ratios. There is some fraction that is still resonant, you get those same effects. But by and large, you've really changed the energy transfer, and you've rerouted energy from here through a different route through the protein. And this has a lot of different effects. And we can do this for all of the different rate constants. We can make sure that all of our model is fully self-consistent. That is, all of the changes are additive, or in the case of spectral densities, multiplicative. You can verify that on this chart if you go through. But as a simple example, if, here's, take this site 3 column. If making this change changes the site energy by 60 wave numbers, making this change doesn't make any change, then doing them both would make a site energy change of 60. It's purely additive. Down here, you oxidize, so you're up to 120. Going to 363 makes a site energy, 353 makes a site energy change of negative 60. This would make a site energy change of negative 100. And 120 minus 160 is negative 40. So everything is still additive. And that's true throughout the model. And what we get then in our model is exactly the same kind of behavior that you might have expected. That say in this oxidized one, you go faster with either mutation, but you then go slower when you do the double mutant. And we see these same types of behaviors. Now, there are some differences. There's some things that we still have to work out. And I think the model itself is actually a little bit naive in some ways. Don't get me wrong, we're proud of it, but it's a little bit naive in that it uses a spectral density, which is an incoherent bath model. It's using a second order perturbation in the system bath coupling. And all of that is doing is allowing the presence of a vibration to drive relaxation. It's not actually allowing a quantum mechanical coupling, so the exciton can split into a vibration and a lower energy exciton. So there's no parametric down conversion or anything like that here. So your real sense of quantum effects is not included in this model, but that's kind of the obvious next step, is to think about this in a little bit more depth and understand how this works. And this is where I think Alexandra's work in particular I would call people's attention to, where she has really thought deeply about the quantumness and the role of these coherences in some of these relaxation mutants, specifically for some of the systems that were looked at by Elisabetta and Greg Scholes. So, there is a lot of theory out there, and we can begin now with an experimental system to sort this out, come up with testable hypotheses, and look at how oxidation of a single residue inside a protein can redirect the energy through two different routes, or can balance the two pathways. We can start thinking about interference between the pathways. There are a lot of things that we can do if these are coherent relaxation mechanisms, and there's some reason to believe that they are. And so I want to leave you with this picture of what's happening inside the protein, where we now, as spectroscopists, can see the transitions only in the chlorine rings of these green bacterial chlorophylls, but we can start with the molecular biology to think about the environment around it, the part that's actually evolved. Because the engineering here is not in the chromophores, it's not in the active molecules. The engineering is in the environment around it. The evolution is in the environment around it. And we can start to look at these single residues, what they do, how they change the energy transfer, and we can use those with a simple experimental handle, that is oxidative stress, to understand how we can manipulate energy transfer dynamics on the femtosecond timescales. And with that, there's a lot more work to be done in the lab. My students are back in the lab working hard right now. And in fact, this particular student is at Intel doing some related work, but I figured for a processing conference, he would be a good person to highlight. And with that, I want to thank the people who are in the group who did the work. It is their hands on the knobs, not mine, especially for the story that I talked to you about today. Jake Higgins was my lead student on that. And Bob Blankenship and his group were absolutely necessary to help us with this genetics, this culmination of a 30-year project to get genetics in these organisms. Of course, I thank my funding sources and you for your attention. And I'd be happy to take questions.
Okay, thank you very much. Thank you very much. That was a very interesting talk, Greg. So we have, I think, a first question on the chat. And the question is, does the effects change with circular polarization and light intensity?
We are working on that. It's a great question. So with light intensity, right now we are working in a 1 exciton manifold. That is to say, with laser light, our third order effect is truly a third order effect, if that's the nature of the question. If the question is light adaptation of these organisms, that is, what is the environment in which they are grown, which is more of a photoprotection question. These organisms are not known to have any photoprotective mechanisms at all. In fact, this oxidative photoprotection that Blankenship found in the PNAS paper is the first one that has been identified. And the reason for that is they live in very, very low light environments. They are outcompeted in higher light environments. So they are sitting at sort of one, in the worst cases, actually one excitation per reaction center per half hour. For scale, the lifetime of one of these excitations is picoseconds. So the chances of having two of them reach the reaction center at the same time is just vanishingly small. So light intensity of the organism, we don't have to worry much about with these particular bugs.
Okay. Would you comment about circular polarization?
Yeah, no, this is a very good question. So chiral effects, anytime one thinks about coupled excitonic systems, you can use chiral spectroscopies to pull apart what's happening. And we anticipate that we will be able to get much more information with two-dimensional chiral spectroscopy. We are in fact doing that in the lab right now. So that's one of the ongoing experiments. I can't really answer the question yet, but that is what's running. We're looking at both the differences between left and right to get a circular dichroism type spectrum, as well as what happens when we use left or right-handed light in various combinations. And with a third order spectroscopy and 4 pulses, there are actually a lot of combinations that you can play with. So there's some that are akin to circular dichroism, but there are actually many more complex ones that one can look at, especially for coherent relaxation, if there's a memory effect of the handedness of the light.
Yeah, you're probably the only one capable of doing 2DES with circularly polarized light.
Some of the experiments were hard, and some of them actually aren't that hard. So I would counsel people to think carefully about them. There are a lot of these that are fairly straightforward. We just submitted a paper on some two-dimensional materials, transition metal dichalcogenides. And there, if you use a left-handed excitation and a right-handed probe, you can actually look at intervalley coupling, and you can begin to look at some really interesting dynamics in the coherences for spintronics without making it a subtraction of large numbers. So the CD stuff is really hard, but some of the others, there's some clever ways to go about it. And some of them are not hard.
Okay. Is there any other question from the audience? Oh, yeah. No. Now I have a question myself, if we have 5 minutes, yes. So can you comment a bit more about how do you expect the quantum effect can affect that table, right, that you showed us, the comparison between the experiment and the red field calculation. So how do you expect that quantum coherence can really make a difference there, right, in explaining the experimental data?
Yeah, so no, it's a very good question. So what I showed you are relaxation rates, and they're sort of agnostic of microscopic mechanism and the nature of that. So a lot of it is somewhat phenomenological, and we should be very honest about that. But when you're building a new system from the ground up and you want to make your own, you can't rely on that sort of phenomenology. And this has been true in photosynthesis for a long time, that when people saw a very fast rate, an anomalously fast rate, they simply plug in an anomalously fast rate constant and everything matches. But that doesn't actually help you build a system. And so one of the ideas that came out of that Plenio and Huaga paper that I was highlighting is the idea that if you have an energy gap that's somewhat small, but you have a vibration that matches it, it's very easy to relax from one state to another and slough off the excess energy in a dissipative fashion to that vibration. And what that means then is you started out with a single particle, an exciton, and you end up with two particles, a new exciton and a vibration. And that can happen in an incoherent fashion, which is simply IVR and dissipative dynamics, which is what's modeled in our red field system. But for a student of quantum, you also realize that this can happen in a much more complex way, where that vibration is either going to mix in a serious sense where you actually get an entangled pair, if you will. It might be a strong word for this context, but a new exciton and a vibration. Or you get something that's actually nonlinear in the sense of McKean. So you have a linear Hamiltonian, linear differential equations that govern the system, the Schrodinger equation. But if you spawn a vibration and that vibration actually starts to affect other electronic states, then suddenly you can see the emergence of coherence in unusual ways and get non-classical behavior. And this is something that Alexandra Alea-Castro has thought about in great detail. And interestingly, I didn't talk about it here, but we published a paper in Chem a few years back where we can monitor the spectral diffusion, or in this case, spectral motion. And what we find is the different states don't move independently. They actually move in sync after excitation. So when you excite to a Franck-Condon region, the motion out of that Franck-Condon region drives concerted motion in all of the other bacterial chlorophyll excited states. Partly that comes by the coupling, but partly that seems to be an effect of the particular protein complex. And so there's reason to believe that when you spawn a vibration in these systems, there's feedback into the electronic dynamics. It shouldn't just be gone and dissipate and be immediately moved on the picosecond timescale far from the electronic dynamics. And so I think these are the routes where we're going to start to see real quantum effects and be able to use coherence as a tool to probe whether there is such feedback in these systems and what the fate of the coherences created by our laser actually are under these environments.
Thanks. I also have another question. What other models are you considering to go beyond the limits of second order perturbation, I guess, in the coupling with the environment?
It's a very good question. So there's a whole family of very common second order perturbation theory models, including red field theory, Forster theory, and the like. To get beyond that, there are some models like the hierarchical equation of motion that allows you to be exact in both the system bath coupling and the electronic coupling, but it still doesn't allow a quantum bath. So there are some models, say some density matrix renormalization group models that can be really quite precise in this regard. But you know, we are a little stuck on this. I mean, I think to answer the question perhaps bluntly and in not the way that the question was intended, we don't necessarily plan to refine this beyond what we have done. We are an experimental group. So we are very much looking for help in doing this. So we want to see it done. We think it's important to be done. But my students are going back into the laser lab, and we're going to tackle, I think, a different suite of questions. So one of the reasons I like talking to a community like this with so many theorists is because I think some of this data demands much deeper theoretical analysis than what we are immediately capable of doing. We're happy to consult. We're happy to collaborate with people. I'd welcome conversations, but we don't actually plan to go back to the computer and try to refine this further. I think that's something that has to be done. And I think there will be more data coming out in the coming months and year that will help us do that more efficiently.
Yeah, indeed. I think this question came from, I think, a theory student.
Oh,
so
it's a great question. It's a great question. And there are two ways to interpret it. One is, does this need to be done? The answer is absolutely yes. The other question is, are you doing it? The answer is actually no. So it's a great place for a theory student to, get their teeth into this problem.
Thank you very much for your very honest point of view.
Well, thank you very much for the invitation, Elizabeth. I appreciate it.
Is there any other question? I mean, we are supposed to go into the round table room now, but if there is another question, we can take the opportunity already from here. Let me check the chat. Okay, so I guess we can have, we thank again Greg for this very nice talk and very nice idea, very nice, exciting idea to how to proceed with the work on this spectroscopy and then quantum biology issue. And I invite you all to join now in 5 minutes the round table event room, where we can kind of wrap up the discussion we had during the whole session and maybe have some more questions for the speakers that can join us in the next room. Okay. Thank you very much again, Greg. It was a real pleasure to have you here. Very interesting. Thanks. Okay, I'll see you all in the round table room. See you there.